Listen-only mode.
Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled ADME 101, Lessons on Running a Successful Clinical Mass Balance Trial. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. Now, at this time, all participants are in listen-only mode. Please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. For 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety, tolerability, pharmacokinetic, and pharmacodynamic studies in highly controlled clinic environments. These include first-in-human dose escalation, drug-to-drug interaction, cardiac safety, bioequivalence and bioavailability, metabolism, and excretion studies, as well as pharmacokinetic evaluations in patients with impaired renal or hepatic function. Celerion offers feasibility, data management, biostatistics, clinical monitoring, and bioanalytical services. Now I'd like to introduce our speakers for today's event. Dr. Foster is the pharmacist in charge at Celerion's Lincoln, Nebraska site. He is responsible for overseeing all ADME studies from a pharmacy, licensing, and total radioactivity reporting perspective. She has worked on nearly 100 ADME studies with all varieties of formulations and challenges. Dr. Foster graduated from the University of Nebraska Medical Center in 2001 with a PharmD. In 2016, she completed the Authorized Nuclear Pharmacist program from Purdue University. She joined MDS Pharma Services in August of 2001 in the pharmacy as a clinical research pharmacist. She then transitioned to Celerion in 2010. Mr. Lester is responsible for overseeing the Celerion staff that manages the execution, compliance, and quality of clinical trials run at Celerion's Lincoln, Nebraska site. In addition, for ADME clinical trials, he is responsible for the laboratory where samples are analyzed in real time to determine radioactivity concentrations and determination of participant release criteria. Mr. Lester graduated from the University of Nebraska-Lincoln with a bachelor's degree in biological sciences. He joined MDS Pharma Services in 2006 as a clinical conduct technician / phlebotomist and transitioned to Celerion in 2010. Previous positions include clinical study manager, clinical conduct supervisor, and bioanalytical analyst. So before we get started, we do have a poll question for audience members today. This is appearing now on your screen, and you can interact with it in real time. The question that we want to ask you before we get started is how many in the audience have planned or executed a mass balance study? Your answers are very simple, just you either have or you have not. Please take some time to click on the appropriate answer as it applies to you or the company that you're working at. The question once more is just looking how many of you in the audience have planned or executed a human mass balance study. It looks like most of you have voted, so thank you very much for doing that. Now let's take a look at the results of that poll. So coming in at 50%, you have not. 42% though, you have. Thank you very much for your participation. Without further ado though, we will pass this now over to Jennifer. Jennifer, you can begin whenever you're ready.
Okay, today we will cover the logistics of ADME studies, an overview, as well as getting into some of the details of running a successful radio-labeled clinical trial. We will discuss the steps needed to start an ADME study with Celerion at our clinic located in Lincoln, Nebraska. We will cover the need for a human dosimetry report and work through the challenges of radio-labeled API, both having it produced as well as the challenges involved with compounding a dosing formulation. We will cover the specifics of Celerion's radioactive materials license, and the regulatory requirements through the state of Nebraska. Finally, we will demonstrate how a successful study is executed by first running a mock preparation, confirming radioactive doses for each subject, and ensuring all samples are collected and data is reported accurately. The overall goal of an ADME study is to see what goes in and what comes out and how that happens. The 4 endpoints are absorption, distribution, metabolism, and excretion. Absorption is the movement of a drug into the bloodstream from its site of administration, often via the digestive tract, before being taken up by the target cells. Factors such as poor compound solubility, gastric emptying time, intestinal transit time, chemical instability in the stomach, and inability to permeate the intestinal wall can all affect the extent to which a drug is absorbed after oral administration. Absorption critically determines the compound's bioavailability. Drugs that absorb poorly when taken orally can often be administered by another route, such as IV or subcutaneously. Distribution is carrying of the compound to its effector site, most often via the bloodstream. From there, the compound may distribute into the muscle and organs, usually to differing extents. After entry into the systemic circulation, either by intravascular injection or by absorption from other various extracurricular sites, the drug is subjected to numerous distribution processes and tend to lower its plasma concentration. Metabolism is the breakdown of compounds as soon as they enter the body. The majority of small molecule drug metabolism is carried out in the liver. As metabolism occurs, the initial or parent compound is converted to new compounds called metabolites. When metabolites are pharmacologically inert, metabolism deactivates the administered dose of parent drug, and this usually reduces the effects on the body. Metabolites may also be pharmacologically active, sometimes more so than the parent drug. Excretion is the removal of the compound and metabolites from the body. There are 3 main sites where drug excretion occurs. The kidney is where products are excreted through urine. Biliary excretion or fecal excretion is the process that initiates in the liver and passes through to the gut until the products are finally excreted along with feces. The last main method of excretion is through the lungs. Celerion has had a radioactive materials license since the early 1980s. We have a team of dedicated subject matter experts ready to help with the unique ADME study design. We have a nuclear pharmacist on staff, a radiation safety officer, and a dedicated ADME lab manager to ensure a successful clinical trial from start to finish. We can provide services of reviewing and or writing your human dosimetry report and your protocol. We will work with you on your dosing formulation, execute your clinical trial, and report your data in order to meet your timelines. The clinic, the pharmacy, and the liquid scintillation counting lab are all located in the same building, which eliminates a variety of shipping delays. We have a dedicated ADME team, which works 7 days per week during your study to ensure daily data delivery. Celerion has executed over 79 radio-labeled studies and enrolled over 538 participants. ADME studies are very unique, sometimes only run once by a client, and we are here to help you through this process. So why would you conduct an ADME study? First, this type of study is required for a new drug application unless there's some reason or agreement that the study is not needed. This study, along with mass balance studies and toxicology species, provides information on the exposure of the parent compound and metabolites. The goal is to understand the full clearance mechanism of the drug and its metabolites in humans, as well as matching the human metabolite profile to what is observed in the animal studies. From this, we will gain the primary mechanism or mechanisms of elimination and excretion from the body, as well as the proportion of parent drug converted to the metabolites. There are 2 main FDA guidances that govern ADME studies, which are listed here. The guidances focus on metabolites and disproportionate metabolites. Disproportionate metabolites are those metabolites that are identified only in human plasma or metabolites present at higher levels in humans than in any of the animal test species. and the links are there for future reference. Generally, metabolite levels greater than 10% of total drug-related exposure at steady state can raise a safety concern. FDA guidance has a decision tree on what to do with those metabolites. We generally will characterize the PK in phase 1 type studies of those metabolites representing more than 10% of the parent. Of course, there's always exceptions to these rules. At this point, we're going to go to the second polling question.
Yes, thank you very much. So once more, we have a poll question that should be on everyone's screen right now. And as before, please take a moment to think and answer as appropriate. The question that we're asking right now is, what is your typically preferred timing for human mass balance studies in an NDA program? Your options are early phase 1, after POC, or phase 2. Once more, the question is, what is your typically preferred timing for human mass balance studies in an NDA program? We'll give it just a few more seconds for everyone to answer. Perfect, we have the majority of you in. Let's take a look how you've answered. Wow, pretty even across the board. 34% at early phase 1, 34% at after POC, and 32% phase 2. Thank you very much for your participation. Back to you, Jennifer.
All right, thank you very much. And really, that's kind of what we see here too. There are definitely varying opinions as to when an ADME study should be conducted, and the answer depends on the overall goals and strategies of the company. One point that everyone seems to agree on is that the study should be completed prior to the initiation of the phase 3 clinical trials of the compound in question. We often see these trials run early in phase 1 when there's been extensive preclinical ADME work completed and there is radio-labeled compound remaining from the studies. Alternatively, we have conducted the ADME study during or after the proof of concept study when the data looks good and it appears the drug is going to continue on and justify the study. The risk in waiting until this point is the availability of the radio-labeled compound. Planning for human ADME studies should be considered during early phase 1 drug development. The planning could be as simple as talking to vendors about radio-labeling your drug, which is often rate limiting in these studies. Identifying metabolites earlier will help to develop your drug metabolite profile. For example, to support your selection earlier in your program. This is a little bit of talking a little bit about the different phases and times that you can run the study. There are 3 prerequisites that are needed before the ADME study can be executed. These include the animal mass balance study, referred to as the QWBA, the completion of the human dosimetry report, and the availability of the GMP-like active pharmaceutical ingredient, referred to as the API. QWBA is a required study in animals prior to conducting the human ADME study. This study is typically completed in rodents and evaluates the tissue distribution of the drug. A QWBA study is used for dosimetry calculations that determines the allowable radio-labeled dose for a human ADME study. The information from the QWBA is used to create the human dosimetry report, which is a report that predicts human exposure from the radioactive dose. The final prerequisite, and one that by far causes the most delays, is the availability of the radio-labeled drug. This should be considered early in the program, and vendors should be contacted to ensure that your timelines will be met. The activities of the typical ADME study include planning, drug prep, conduct, data management, and reporting. Stage 1 will include finalizing the protocol and ensuring the human dosimetry report is complete and acceptable. We can provide both protocol writing and dosimetry writing if these services are needed. Stage 2 will include drug prep and dosing solution verification, with a mock prep taking place at least 2 weeks prior to dosing, and the actual prep taking place on the day of dosing. During the conduct stage, subjects are in-house, samples are being collected and analyzed daily, with results being reported to the sponsor every 24 to 36 hours to ensure timely release of the participants per protocol. Subjects are housed in a dedicated conduct area that is maintained per our radiation safety SOP. The final stage will include final recovery data, a total radioactivity report, case report forms, and the PK statistical analysis. The typical ADME design includes 6 to 8 healthy male subjects with carbon-14 as the most common radionuclide. Compounding is almost always required, either with a hot and cold drug separately or a blended material compounded into a final dosing formulation. The 4 most common matrices include whole blood, plasma, feces, and urine. Our typical release scenarios are listed here, with standard confinement ranging from 7 to 14 days and recovery ranging from 85 to 95%. Our longest confinement here in Lincoln has been 49 days for an ADME study, with our longest surveillance being 9 months. Here we have provided a visual timeline of how these pieces fit together for an ADME study to dose on time. Again, as I've mentioned before, the drug is typically the rate limiting step. Celerion's radioactive materials license is issued through the state of Nebraska. The license is amended with every compound that is tended to be administered to human subjects. The amendment process typically takes 3 to 4 weeks after IRB approval. There are 5 things that are needed for amendment to be processed, and these include the IRB approval letter, the final protocol, the informed consent, the IND in effect letter, and the human dosimetry report. Once all of these documents are ready, we will submit the study to the state for license amendment. Celerion follows USP 795, 797, and 800 for pharmacy compounding. USP 795 focuses on non-sterile pharmaceutical compounding, which would include capsules, oral solutions and suspensions, and any kind of topical preparation. USP 797 revolves around sterile preparation and would include IV preparations, subcutaneous preparations, and inhaled preparations. Celerion has the ability to perform low, medium, and high-risk sterile compounding. That means we can perform simple aseptic manipulations of all sterile products, as well as start with non-sterile active pharmaceutical ingredient. When we start with non-sterile API, we will put it into a solution, perform sterilization with a 0.22 micron filter, and administer the product as a sterile product. In the case of a non-sterile to sterile preparation, we would have a sterility test performed on the mock preparation by a contracted lab and perform an endotoxin test on site. On the day of dosing, the formulation would be prepared in the same manner as the mock preparation, and the on-site endotoxin test would again take place, and additional samples would be sent for sterility to confirm what was administered on the dosing day was sterile. USP 800 is a new regulation in the USP that is not yet in effect due to appeals. However, the intent is to protect the worker from exposure to hazardous drugs. Most phase 1 API would fall into the hazardous drug category because the information simply is not available in the early stages of research. There's a variety of equipment that is needed to meet the requirements of USP regulations as well as successful formulations. The equipment includes biological safety cabinets, balances, stir plates, sonicators, vortex mixers, and other equipment as appropriate per protocol. All Celerion's equipment is maintained per SOP and verified by a third party to ensure proper working conditions. All investigational product is stored in temperature-controlled areas, including room temperature, -20 and -80 centigrade. Refrigerators and freezers are on backup generator, and pharmacy staff is on call 24/7 to ensure proper storage of investigational product. This slide is a layout of our new compounding suite, which opened in February of 2020. The ADME suite is blue in this layout and is a negative pressure room and vented to the outside. The room contains a powder hood for non-sterile compounding and a BSC for sterile compounding. There is also a refrigerator, a -20 freezer, and a -80 freezer. The rest of the compounding suite is for other types of compounding, including hazardous sterile compounding, non-hazardous sterile compounding, and hazardous non-sterile compounding. All rooms are certified to USP standards every 6 months by an outside vendor. Pharmacy staff are also trained to USP standards and are tested by written testing, gloved fingertip sampling, and media fill testing every 6 months. The compounding process at Celerion starts with the client supplying information on the compounding procedure to the nuclear pharmacist. A master formulation record is then created. Once the sponsor has approved the master formulation record, the mock preparation can be performed. Any modifications that need to be made will be documented and updated during the mock prep. The mock prep is typically very informational and helpful, so the day of dosing is seamless. Compounding for the actual study typically takes place on the morning of dosing, usually very early, due to the length of the various compounding procedures, as well as the need to verify the radioactive concentration prior to dosing. Our pharmacy staff is able to receive radioactive compounds accurately and safely. We have processes in place to ensure there is no contamination, including a dedicated receiving area. All radioactive shipments are swiped outside and inside to ensure there have been no spills during transit. All investigational products as well as excipients will have accountability records. The lot number, expiration date, receipt quantity, certificate of analysis, safety data sheet will be tracked, as well as any other relevant information. All products are received by 2 pharmacy staff members. We are able to ship or destroy any remaining investigational product or excipients at the conclusion of the clinical trial. As mentioned, we will amend our state license per compound and also have a radiologist on call for all ADME studies. Our on-site pharmacy services include the dedicated compounding suite for ADME studies that we mentioned in the layout. The ADME suite is an area that is swiped for radioactivity pre and post compounding per the radiation safety SOP. This is where the actual compounding will take place. All compounding at Celerion involves a pharmacist, in the case of the ADME studies, the nuclear pharmacist, and another pharmacy staff member. The compounding is documented on the master formulation record and sent to the sponsor when complete. We will always either verify the certificate of analysis of the radioactive API or the concentration of the final dosing solution. Typical sampling of the solution or suspension includes sampling in triplicate of the top, middle, and bottom of the bulk solution. Additional sampling is always an option, especially when dosing an IV study, and there's multiple IV components being used, such as tubing, filters, and catheters. All radioactive doses are measured by weight, so the exact dose is known per subject after determination of the concentration. If possible, we will also measure residual radioactivity in the dosing container and subtract that from the dose, making the measurement even more accurate. The most common formulations that we see for ADME studies include capsules, solutions and suspensions, and IV formulations, with solutions and suspensions being the most common overall. Every formulation is different and has its own challenges. We have a lot of past experience to ensure we will end up with a successful formulation at the end of the day. Here's a look inside of our dedicated ADME compounding suite. The powder hood for non-sterile prep is in the top photo. Within the hood, we have a 5 and a 2 decimal balance equipped with printers. We have a variety of positive displacement pipettes for sampling. We also have a heated magnetic stir plate. The bottom photo is our BSC, which is used for sterile ADME work. As mentioned, this room is a negative pressure room certified to USP 800 for the safety of the worker. And Ryan, we're now ready for polling question number 3.
Excellent. So, as everyone should see on their screen right now, the question we have for you is, when do you expect to execute your next human mass balance study? The options available for you are this year, over 1 year away, or over 2 years away. Please take a moment to answer as appropriate, of course. Your options, the question again is, when do you expect to execute your next human mass balance study? We'll just give everyone a moment to answer. A few more seconds. Perfect. Looks like everyone, the majority of you have answered. Let's take a look at your results. At 41%, you have said over 1 year away, 32% over 2 years away, and then 28% this year. Thank you very much for your participation. You may, this is back to you, Jennifer.
All right, at this time, I'm going to hand it over to Brandon.
Okay, can everyone see my screen okay?
Yes, we can. It looks great.
Thanks, Ryan. Well, first of all, I'd like to thank everyone for joining the call today. It does mean a lot to us that you took time out of your busy schedules to attend. And I hope that everyone is staying safe and healthy during these times. So going back to the polling question, it's great to see that a number of you on the call are planning an upcoming ADME study. We would certainly love to work with you, so please consider Celerion. Jen or myself are available to answer any questions you may have via email or a phone call, and we'll give you that contact information at the end of the presentation. We'd also love to have you out for a site visit when that is appropriate to do so, and discuss your study in more detail and tour the facility. For those of you that might not have a planned ADME study upcoming, do keep Celerion in mind for your other clinical trials. We are a full service CRO, and again, happy to discuss any of your upcoming clinical trials and see how we would be able to help you out. So over the next few slides, I'll be covering the logistics of sample collection, sample analysis, and data reporting to ensure that we conduct successful ADME trials. So the first step in the planning phase is to draft the total radioactivity analysis plan, or we call it the TRAP for short. So that's drafted by myself at around the same time that the study protocol is finalized. So this manual outlines the procedures for pre-study urine testing, which we'll talk more about, and how urine, feces, plasma, and whole blood samples for total radioactivity are collected, processed, and analyzed, and those results reported. So the document is created according to Celerion standards and then shared with the sponsor and also labs that may be involved in subsequent analysis, such as the metabolite profiling laboratory, for review, and then we'll customize it from there. So we customize it to each protocol based on the compound information and that feedback that we do receive from the sponsor and other reviewers. You know, one example of customization would be working with the sponsor and the metabolite profiling lab to ensure that the fecal homogenization solvent that we use is acceptable for the metabolite profiling work and other sample analysis from the fecal matrices. All right. So the next pre-study activity to take place is a urine adsorption test. So from our experience from working with these radio-labeled compounds, we've found that many of them with high protein binding affinity will bind to the walls of our urine collection containers. So this is a critical pre-study test to conduct, since any significant binding can lead to under-reporting of overall recovery, especially important for compounds that have a significant portion eliminated through the urine. So this test is conducted using the mock dosing formulation that Jen had talked about. So for this testing, we will spike urine samples and store them in the same containers that urine will be collected in during the study. Our standard is to test samples of urine alone, urine with 0.25% tween 20, and urine with 50% methanol, as from our experience, these have been proven to be effective to prevent that binding. So we'll initially analyze the samples and then store them refrigerated for 24 hours to simulate a 24-hour collection interval, and then they're reanalyzed to determine if there is compound adsorption. We'll summarize those results and provide those to the sponsor to determine if an additive is needed during collection in the clinic. So one important thing to note with this testing is that we always will have a discussion prior to the testing beginning to confirm that the additives being tested are acceptable for the subsequent sample analysis work. So we commonly do modify the additives tested or test a different additive concentration based on these discussions. For example, we may want to test an additive due to a known urine metabolite that requires stabilization. All right, so moving into study conduct, we do have dedicated confinement areas for our participants and dedicated sample processing areas for handling radioactive samples. Prior to the study, the dedicated station and all equipment used is swiped to verify the areas free of radioactivity according to our SOPs. The restrooms are set up with commodes and the toilets are locked and inaccessible to the participants to ensure that all urine and fecal samples provided are collected. Upon admission to the clinic, the clinical study coordinator will orient the subjects to the facility and educate them on the dosing procedures that will take place the next day, and also educate them on the sample collection procedures and the importance of collecting all urine and fecal samples during confinement. So during confinement, during the study, the restrooms remain locked at all times, and we do schedule an associate a 24-hour sample collection and compliance monitor to be present in the conduct station. So if a participant does need to use the restroom, they must check in with the monitor first, and they're provided with the necessary sample collection containers. Urine and fecal samples are collected electronically using Celerion's proprietary e-source. It's named ClinQuick. So ClinQuick is a purpose-built system that we've developed in-house for data collection for phase 1 studies in particular. It does have built-in logic checks within each module to ensure data is collected accurately and error-free the first time. So just to kind of walk through in a little more detail how samples are collected using ClinQuick, when a participant needs to use the restroom, their armband is scanned and then a collection container with a barcode is scanned and checked out to that participant, and they're let into the restroom. And then when the sample is returned, it is scanned back into ClinQuick to check in the sample. Pooling is all done electronically as well. So when the samples are pooled, there's a pooling container assigned to the participant, which is scanned, and the collection container is scanned, and ClinQuick verifies as a double check that the containers belong to the same participant and same interval to prevent any sample pooling errors. So that's a good example of some of the built-in logic checks that we've placed into the system to ensure data is collected accurately. There's also data collection modules for dosing, blood collection, meals, adverse events, just to name a few. But a majority of the data collected at our at Celerion for all of our phase 1 studies is collected primarily electronically using ClinQuick. One benefit of this to our sponsors is the data is available instantaneously via our client portal named Selexis. So prior to each study, the sponsor representatives are granted access to Selexis. And how that portal works is each time you log in to Selexis, the data collected up to that point in time for your study is refreshed and available for review. So you really do have real-time access to the data being collected on our clinical trials here at Celerion. And then at the end of the study, the conduct areas and equipment is swiped again to verify it's free of radioactivity. So laboratory setup, prior to each study, the on-site liquid scintillation counting laboratory located on the 3rd floor of the building is prepared for sample analysis. Pre-study swipes are performed in this area as well to confirm the lab is free of radioactivity. The liquid scintillation counters are calibrated and verified to be operating as expected prior to each study. The liquid scintillation counters are checked daily using a set of normalization standards to confirm that they are operating as expected throughout the study. The sample oxidizer equipment that we use are verified as well prior to the study by performing a recovery and a carryover test. So control samples, oxidized samples, and Blake samples are tested, and the oxidized samples must be within 3% of the expected value, and carryover between samples needs to be less than 1%. Our balances that we use during sample analysis are verified on a daily basis, and we use those balances to document the fecal homogenate weights and individual sample aliquot weights. And then blank, low, and high quality control samples are prepared and characterized prior to each study. These QCs are included on each clinical sample batch to confirm batch acceptance. So now moving into a little more detail on how samples are analyzed and results are reported. At the end of each collection interval on a daily basis, samples that are needed to determine release criteria are transported upstairs to the lab for analysis. We do have a dedicated and specially trained team of analysts that perform this work on our ADME studies, and the lab operates 7 days a week when these studies are in progress. So priority is given to urine and fecal samples, as these results are most commonly needed to determine if participant release criteria has been met. Typical participant release criteria is recovery greater than 90% of the administered dose, or less than 1% recovery in 2 24-hour intervals where both fecal and urine samples were provided. Sample analysis is done by weight, and samples are typically analyzed in duplicate, and we do things by weight, so it's as accurate as possible, and recovery calculations are as accurate as possible. So how urine is handled: urine's directly aliquoted into counting vials, and each aliquot again is accurately weighed. The liquid scintillation counting cocktail is added to the vial and mixed, and then those samples are placed directly onto the counters. Recovery is then calculated for the interval based on the DPMs per gram in each aliquot and the total volume of urine provided from that interval and the total dose administered. Fecal samples are a little bit more involved. We do have to first homogenize them, and we need to add a solvent to facilitate that homogenization. So we add 4 times the sample weight provided during the interval. And a typical homogenization solvent would be maybe a 20% ethanol solution or even just ultra pure water. We do prefer to include an organic solvent in there, such as ethanol, as that does tend to produce a more homogeneous sample. That solvent to be used for each study is discussed with the sponsor and the metabolite ID lab to confirm that it will be acceptable for subsequent sample work. So after homogenization is complete, the homogenate is aliquoted, and each aliquot is weighed. Aliquots are dried, oxidized, and collected into counting vials and then placed onto the counting machines. So then we'll back calculate recovery based on the DPMs per gram in each aliquot, the total weight of the homogenate, and the dose administered. So plasma samples are handled in the same manner as the urine samples, typically not included in release criteria. So they would be batch analyzed throughout the study and finished up at the end of the study when the last participant's released. Whole blood is similar to fecal sample analysis, just without the homogenization step. They're aliquoted, oxidized, and then counted. And those again are batch analyzed typically throughout the study and finished at the end of the study. Should an occurrence of emesis occur during the study, we would notify the sponsor immediately. We will analyze that sample using the same method as the fecal samples to determine the amount of dose that was lost. Then we discuss with the sponsor if it's appropriate for that participant to continue in the study based on when that took place. All of this data is QC'd in real time, and results are reported to the sponsor on a daily basis. There's an approximate 24-hour lag from when that interval ends to when those results can be provided the next day, just with all the analysis activities that need to take place. Batch acceptance, batch and sample acceptance criteria. So like I said, we include those QCs on each batch. The blank QCs need to be less than 100 DPM, and low and high QCs must be within 20% of the expected value. If these criteria are not met, the batch would fail and we would reanalyze the batch. Clinical samples are typically run in duplicate and must be within 20% of the mean value of those replicates. So if that criteria is not met, the samples fail and they would be reanalyzed on a subsequent batch. All right, so moving into a few case studies, we have run a number of these ADME studies here at Celerion, and would like to share a few of our experiences with everyone today. So, more often than not, everything goes as planned. The dose is administered, samples are collected, we hit that 90% recovery threshold, and participants are released. But occasionally things don't go quite as expected. So homogenization problems and solutions. So some compounds can be a bit tricky to work with during homogenization based on their characteristics, and Jen usually experiences these problems much earlier during her mock preparation and compounding activities. But prior to each study, we will review the route of administration, the solubility information, and the expected route of excretion. Some compounds that have been problematic in the past have been ones where we're administering the compound directly, such as a powder and capsule, without solubilizing the compound prior to administration. In addition, the compound is not readily absorbed, primarily excreted through the feces. So based on the solubility information, we look to select a solvent that's going to assist with producing a homogeneous sample. And if we expect this, we can do some pre-study testing, making some test samples and testing different homogenization solvents to ensure that we're going to have fecal homogenate that is acceptable. So, if we plan accordingly, this isn't an issue, but during the study, we do watch for variability among our duplicate sample replicates and variability in general amongst the participants, as this can be an indicator of homogenization issues. And one thing we do for all of our studies as an extra precaution is to retain all of the fecal homogenate until we're confident that we've achieved acceptable recovery. And if needed, we can go back and do some rehomogenization since we have all the sample retained with different solvents, and we've also used different sample extraction techniques to recover radioactivity from fecal homogenate in difficult situations. Low radioactive doses. A typical radioactive dose for a human ADME study is 100 microcuries. However, they can be higher or lower based on the compound. We're seeing a number of protocols now, including an absolute bioavailability arm with a low-level radioactive IV dose. And these are kind of in the microdose category, around 1 microcurie. And then after that, dosing has been performed and a washout is confirmed, a traditional oral ADME dose is administered. So we have successfully analyzed urine and fecal samples from doses as low as 1 microcurie and achieved 90% recovery from those low-level doses prior to moving into those traditional oral ADME doses. So in order to quantify those samples, we need to lower the limit of quantitation, and we do this by increasing sample volume and lengthening the count time on the liquid scintillation counters. Then extending confinement. This has happened on a couple of occasions. So it's most important during protocol design to predict the maximum confinement based off the PK information available for the compound. And we have a team of protocol writers and statisticians that can assist with this prediction if needed. But a typical maximum confinement is between 14 to 28 days. More often than not, our studies are released within that maximum confinement period, but on a couple occasions we have seen recovery move a little slower than anticipated and extended confinement to ensure we achieve 90% recovery. So we're looking at data on a daily basis with the sponsor, as I mentioned, and when we think that we may not quite get to 90% and extending confinement for an additional week or a period of time may be necessary, we're able to work quickly internally and with the sponsor. So working with our protocol writing team to get an amendment in place, ICF updated, and submitting to the IRB. We'll keep our participants informed of the situation as well and let them know about the possibility of extending confinement. We find that in these situations, participants are generally agreeable to extend confinement since this results in additional stipend. But in the event a subject can't accommodate a confinement extension, we work by sending home collection supplies and instructions, and they'll collect samples at home and then return to Celerion for analysis to further characterize the recovery in those situations. And with that, I'll turn it back over to Jen to wrap up the discussion.
All right, thank you, Brandon. So at the end of the day, the deliverables that we will be providing include the recovery data that Brandon mentioned, which you'll be getting an ongoing report throughout the study, and then the final data at the end. There will be a total radioactivity report, which is written by myself, which is basically all of the data put together and a summary of the dosing. And typically that is amended or as a supplement to the CSR. Other services that we can help you with are listed here. We can help you early on to make sure that your drug is radio labeled and received in the pharmacy in a timely manner. We can provide you referrals for the QWBA studies if you need that done, and we can also help you with a human dosimetry report. We can write that and review it and help to make sure that the reporting is accurate for the state submission. Finally, we are able to help you with metabolite profiling, either by referral or in the near future, our facility in Zurich, Switzerland. Our bioanalytical facility in Zurich, Switzerland is currently in the process of setting up and qualifying their metabolite profiling laboratory. In the very near future, this service will be able to be performed under the Celerion brand, and we're very excited to be rolling that out. At this time, I would like to thank you for joining us today. We take pride in making our clients feel welcome here in Nebraska. Please feel free to reach out to Brandon or myself at any time on questions on today's presentation or any of other Celerion's capabilities. We will now open the lines up for questions.
Well, thank you very much for that insightful presentation. I would like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. Now, I've already received some questions, so let's get started with those. Our first question we have asks, does it have to be male subjects? Why not have female subjects?
So I can go ahead and start with that. Typically, it can be non-reproductive females. So we have done that before, but for the most part, it's easier as far as collection is concerned. As far as urine, feces, you don't need to worry about menstruation, that kind of thing. But if they are non-reproductive females, that is something that's possible. But overall, we have just done male participants.
All right, thank you very much for that answer. Our next question asks, what is your experience in allowable radioactivity between the US and UK?
So the UK, their exposure limit is a little bit less than ours. So we follow the CFR guidelines. And so ours is just a little bit higher. And so as long as the dosimetry and the human exposure is okay, then that's what it is. So typical 100 microcurie and the 300 millirem for the year. So that's why we need that human dosimetry report written to ensure that the exposure is not over the allowable US limits.
Thank you very much. Our next question asks, do you need a dosimetry report from RAT QWBA if you are planning to perform human radiotracer dose study?
So our license is a blanket license under the dose of 500 nanocuries. So the answer to the question, if your dose is less than 500 nanocuries, no. And if it's over 500 nanocuries, yes.
Thank you very much. Our next question asks, does Celerion have experience in outsourcing AMS, accelerated mass spectrometry, and the ability to conduct metabolite ID internally?
So the first question, yes, we have definitely done that, shipping samples to AMS vendors. We definitely have done that several times, and we can work with you to do that. And as far as the metabolite profiling internally, we are getting that up and running at our Zurich, Switzerland facility. And we also have preferred vendor contracts that we have qualified and are able to provide referrals on that for the metabolite profiling.
Perfect, thank you very much. The next question would like to know how many beds does Celerion have dedicated to radioactive AME study?
Brandon, do you want to
take that one?
Yep.
Yeah, we our are currently able to run 2 studies simultaneously, possibility to run 3 at the same time, but we're constantly evaluating our services and looking to hopefully expand those capabilities in the future. But so I guess the short answer is 2 studies. We have 200 beds in our facility, so we have plenty of beds to accommodate different participant sizes depending on the ADME protocol.
Thank you very much for the answer. Our next question asks, could the new compounding facility equipment audit and qualifications document be provided to a sponsor for QA review of the changes to previous Celerion qualification?
Sure. So the other thing that we've been doing a lot is a remote audit. So if you wanted to reach out to me, or probably start with me, and I can refer you to the QA group, and we could set you up for a remote audit type situation and provide you with every kind of QA document that you would need.
All right, well, thank you very much for those answers. However, we have reached the end of the question and answer portion of this webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you, or if you have further questions, you can direct them to the email addresses on your screen. I want to thank everyone for participating in today's webinar and remind them that in the handouts tab of the control panel in GoToWebinar, you'll find a document provided by Celerion for you to take home today. You'll be receiving also a follow-up email from XTalks once the event is done today with access to the recorded archive for this event. Also, a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, I'm also going to send you a link in the chat box right now, and with this link, you'll be able to view the recording of this event on that page. You can also share this link with your colleagues. When they register for the recording here, they'll be able to view it there. So I encourage you to do that. And also, please join us in thanking our speakers, Jennifer Foster and Brandon Lester. We hope that you found the webinar informative. Please have a great day, everyone. Thank you for coming.